Each one of us, for sure, has been fascinated by the huge diversity of leaf shape that exists in nature. Leaves come in many different forms and shapes. We in our department are very interested in identifying the genetic basis of different leaf shapes. We work with Cardamine hirsuta in our lab, which is commonly known as hairy bittercrush. It is a small crucifer related to the model plant Arabidopsis thaliana. Arabidopsis thaliana has smooth, simple leaves, whereas Cardamine hirsuta has complex leaves divided into leaflets. As the leaf develops, it undergoes a transition from sink to source. A sink organ is a net importer of photosynthetes, which is synthesized during photosynthesis, whereas a source organ is a net exporter of photosynthetes. This transition from sink to source is not rapid, and it begins at the leaf tip and progresses towards the base. An interesting question that we would like to answer here is whether the sink to source transition is a key physiologically relevant readout of the age-dependent pathway. And also, how do these events differ between the simple and compound leaves of Arabidopsis and Cardamine respectively, which have different leaf morphology? Another interesting feature about Cardamine hirsuta is that within the same plant, over the turn of its life cycle, aging leaves shows increased morphological complexity. This morphological change process is called heteroblasty. Moreover, the natural strains of Cardamine hirsuta from across the world shows considerable variation in leaf length, leaflet number and flowering time. Taking advantage of this variation in leaf morphology and using genetics as a tool, we in our group identified genes controlling age-dependent variation in leaf shape. And previous studies in our group showed correlation between leaflet number and flowering time. Early flowering plant produces more leaflet at the same leaf nodes compared to the late flowering plants. This results leads to our main hypothesis, that is, the coordination of flowering and juvenile to adult phase transition may allow sufficient resource production like sugars and their transport from source leaves to the sink organs like flowers and seeds. The main question that I would like to answer through this is whether the age-dependent variation in morphology is linked to physiological events during plant aging. This study will help us in understanding the physiological and metabolic consequences of diversification of leaf margin complexity in Cardamine hirsuta.